tend to, if we're going with the assessors, we tend to say just purely under
striking me is that there is something called proposal, let's say, number of individuals, something about could be differentiated for different groups of species. Because for lichens could be different, for large mammals also. But maybe there are some studies of populations or genes that prove that.
That's precisely why I've said when they develop the system, they sometimes get studies are that's really looking for it.
Yes. The truth is those
are not listed under criteria C or even D for that matter because their populations are just that's they're also listed under B or D.
Okay.
Sometimes under A, but when it comes to other like marine mammals are almost never listed under or sometimes you can see some of them.
Okay, I think all three groups, or 2.5 groups, have reached a conclusion. So under criterion C, hands up all those that got critically endangered. Under C. Criterion C. Yes, you did, I know you did. Okay, so the assessors of this species also got critically endangered. So we've got currently the wild population is certainly less than the 250 threshold. It's between 60 and 102 individuals. In fact, it may even be less than 60 based on the DNA analysis study that was done more recently. So it certainly meets critically endangered C under population size. Studies have shown that the population has declined by at least 25% within the last 100 years, which is one generation. So that meets 25% in 3 years or one generation for C1 under critically endangered. There is a continuing decline in the number of mature individuals. And also there are less than 20 plants in each subpopulation. The subpopulation in this case is pretty much one individual in each because they're all separated. Individuals are separated by at least a kilometre of distance. And also there's been no recruitment, there's no genetic exchange going on, they're not supporting pollinators, so it's extremely fragmented. Less than 20 plants in each subpopulation, certainly. So it's certainly critically endangered C2A1. No single subpopulation holds most of the individuals. It seems from the information here is fairly evenly spread throughout the subpopulations. So we wouldn't use C2A2, and we don't have any information about extreme fluctuations. It seems unlikely in this case. So this cycad would be listed as critically endangered C1 and C2A1. Anyone get any different? No? Excellent. I'm glad criterion C was understood quite well. It's a difficult one to teach normally. Criterion D. Clearly I've shown the answer there. Did anyone assess this as critically endangered under criterion D? Endangered D. Endangered D. Endangered D. Okay. Yep. That's absolutely right. So I've highlighted an obvious typo here that we have to fix. This is not a fish, this is a cycad. We will fix that for the next workshop. Interestingly, the species also qualifies for vulnerable D2. Did anyone pick up on that? Probably not, because you're looking at the higher category, so you would, under that criterion, you would assess it under the higher category. But if endangered didn't fit, then this would qualify for vulnerable B2 as well, because it's got a very small area of occupancy, about 9 square kilometres. There's only 2 locations estimated for the species. There is a very real threat there, not just a plausible threat, there's a threat ongoing at the moment. The collecting pressure could very easily wipe this species out in a very short space of time. So all of that qualifies it for at least vulnerable D2. However, we already know that it's critically endangered under the other categories, so that's under the other criteria, so that's how we would go with the final assessment. Quantitative analysis. Did any of these 2 groups use quantitative analysis? No?
Only that not that big.
No?
We put as a critically,
Oh.
but we got the idea.
We had
Confusing.
Okay.
discussion afterwards and we realised, we just thought that when they said 30% decline is expected to take place within the next 30 years, and it was a sort of that indicator they must have done some modelling to make that they.
Yeah, same reasoning that Michael did as well. I agree with your thinking. I'm given the information presented in front of you. I agree with that thinking. I think it's very logical to think, given all the evidence that the assessors must have done some kind of modelling, and all the evidence of the lack of pollinators, etc. The actual situation here, which you don't have the benefit of the assessors' minds on this and on their insight and data, they were reluctant to go down that route and say, yes, it likely will become extinct within a certain time period, within 1 generation, which is 100 years in this case, because they do have these conservation actions which they are implementing, and potentially they could actually go around and hand pollinate. There's so few of them, they could actually go around and hand pollinate them, which debatable. Does that mean it's extinct in the wild, really? You know, it's being supported by humans. But they didn't go down that route of quantitative analysis, possibly because they want to keep the conservation work going. You know, they don't want to say, certainly it's not data deficient, but they don't want to say this is on its way out because the funding might dry up.
But I just think a little bit of background, which I told Rebecca, I mean, our organisation was involved in this PHBA at the start, and I remember now coming back to me about how they chose not to do any actual PBA because they didn't have the information that they needed.
Ah, right.
So I don't think there were any models actually run at all.
Okay, so the information that we put in here is actually a bit misleading.
Well, what you've written there is correct when you say no quantitative analysis for that.
Okay. It says here, oh, I see, population PBA was developed, but
See, the that's a PHBA, and there was certainly a report, but as far as I recall, there wasn't any actual PBA done because
Okay.
they just didn't know enough about the demographics of the population.
okay, right, that makes sense. Thank you. Right, I'm not going to give a presentation on the last little bit because it's so straightforward. This is a long drawn out exercise of what you would normally do in a much quicker time period after having the data all gathered together. Look at your data, you go through each of the criteria one at a time, and then to get your final assessment, you go through, you look at the results of each of the criteria, and you choose the highest category, and that is your final assessment. So, in this case, we have, under criterion A, the species would be critically endangered. Under B, it would be critically endangered. Under C, critically endangered. If we just looked at D, it would be endangered. And if we just looked at criterion E, it would be data deficient, basically, because we haven't got enough data to say. So the assessment would be critically endangered, yes. And in that assessment, you would make sure that you cite all of the criteria where the thresholds apply to that category. So here we've used criterion A2, A4, B2, C1 and C2, but we haven't mentioned anything about it qualifying as endangered under or vulnerable under criterion D because the critically endangered thresholds were not met. You could mention that in the text, but it wouldn't go into the final assessment. It's only the criteria that fully qualify for that category. Alright.
It's good that people are see. It's quite lengthy if you put it all. I mean,
It
it's good
is.
that you only put it here.
It's a lot easier just to put CR, yeah.
I was going to ask, I mean, compared to most species, that must be quite a long list of letters.
It is, yeah. It's deliberately chosen for training purposes because it is actually quite involved and makes you think about different criteria all at the same time. Usually, things are assessed under 1 or maybe 2 criteria, but occasionally you get things that are assessed under 3. I don't think have we got anything that's assessed under all 5? I don't think so. I'll be interested to look and see it. I don't think we have it.
I don't think we do this in Parliament.
We probably would have used it exactly, yeah. Are there any more questions about the criteria at the moment? So the user guidelines has much more information about different scenarios, and when you start using your own data, that's when the big problems come up, and you think, in theory this is easy, but actually in this situation, it involves quite a bit of thought. Okay.
What are the species that we don't really know the generation of times, generation of lengths, things like that, the lengths of the age of the...
So where you've got data gaps in
Yeah.
generation, being able to calculate generation length and things like that. And sometimes you will have more data on the range rather than the biology or the population size. So you might not have information to see what the rate of decline is, or even which time period you're meant to be using, but you might have lots of range data, so you may be focusing heavily on criterion B, and your consideration of criterion A is quite cursory because you stopped at the first point. But then if you're dealing with, well, I know you're working with amphibians a lot, and many, I know not all species of amphibians are very short-lived, but some can be relatively long-lived. But in many cases you can actually use similar species from maybe a different genus or something, but better studied, and you can infer that the generation length is going to be very similar. So you can use that in your inference, but just document that in the assessment. Don't just say the generation length is. Say, well, we infer the generation length is this because we don't have data directly, but we do know about this other species which are closely related.
But the problem is in Indonesia with 360 something species. Maybe they well studied only 3 or 4 species.
Yeah.
But then in that case, isn't it useful to try and do a PDA and look at the sensitivity of your model outcome based on how you change generation? So you could say, if my generation length is 2 years or whatever, then how does that change the outcomes if it's individual when it's 3 years? And then you can...
Yeah, but the only thing is with the PDA, you do need a certain amount of other data as well. That's quite a lot. So that's usually the sticking point. If you haven't got this information, you've often not got
Bible of this juvenile and things like that.
Yeah, sometimes you know the species just from the original collection.
Yes.
That's it, there's nothing else. So either it might be data deficient, or you might have evidence of, we know it was collected from this area, we haven't seen it elsewhere, but we know that area is completely trashed now, and similar habitat is being destroyed elsewhere, so you then have to, it'd be a weaker assessment, but you then have to suspect what's happening to the species, or what has happened to the species in some drastic cases. The user guidelines do give some examples of that kind of scenario. What do you do when you only know the species from the technicality and nothing else? And there's different scenarios. You can end up with it being assessed as threatened, or least concern, or in worst case scenario, even critically endangered, possibly extinct. Okay.